up to me.
Maybe you just need to go down?
Yeah.
They will come in.
Yeah.
Cool.
Good.
Hello, hello. Hi, everyone. Okay. Welcome to the afternoon session. So this is the first of our series of 4 sets of contributed talks. We've got an amazing lineup. We've got 6 speakers. They come from many different universities, different career stages, and they're going to talk about different things, but they're all going to be amazing. Each speaker is going to talk for 15 minutes. That slot is going to be used partly for a talk and also for questions at the end, so feel free to use the microphones after each talk. And that's it. So the first speaker is Jennifer Groh from Duke University.
Thank you. How's the sound? Is it good? You can hear? Awesome. So thanks to the organizers and also the reviewers for selecting this work for a talk. I am filling in for some big shoes here. My student, Meredith Schmale, was unable to attend, so I'll be giving the talk in her place. So I'm going to be talking about a really simple and obvious problem, but one that I think has been overlooked in recent years, and that is the question of how the brain represents multiple stimuli. And the reason I think this is an unsolved problem is that receptive fields are actually rather big. Depending on how far away you're sitting, your foveal V1 receptive fields probably contain more than one letter on this screen, and certainly more than one component of individual letters. And yet you're able to resolve the fine details of the visual scene. The problem is basically that populations of neurons that are potentially responsive to each stimulus can overlap. So the letter A here might evoke a response in a distribution of neurons, and the letter R adjacent to it would evoke activity in a different population, a slightly different population of neurons, but that overlap poses a problem. So how does the brain preserve information about multiple stimuli? And this problem gets worse and worse as you progress along the visual pathway and receptive fields get larger and larger. And current approaches to this kind of problem typically focus, in my opinion, on how to be sensibly lossy about coding information. So for example, work focusing on selective attention concerns how does the brain prioritize one stimulus at the expense of others. Work focusing on observations under the heading of normalization in the brain suggests that neural populations effectively compute an average between the stimuli that are presented, and that too would result in the loss of information about each individual stimulus. So the question of how there's preservation about information about multiple stimuli is unanswered. And the proposal we're putting forth is that neurons might multiplex. And this concept comes from telecommunications, where if you have one wire and more than one signal that you'd like to send down that wire, you could imagine chunking those signals across time and interleaving them, and thus providing information about both signals. This is called time division multiplexing in engineering, and we've been interested in whether or not there is some version of that fundamental concept that might be applicable in neuroscience. The first data set that I'm going to tell you about involves area MT, and specifically the face patch system. And this data comes from Vinrik Frywald and was collected by his graduate student, his former graduate student, Ekinora Ebahara. The interesting thing about IT is it's one of the places with the very largest receptive fields. However, it also has selectivity for faces as compared to non-face objects. And I think you can think of these two forces as, these two factors rather, as working in opposite directions. The large receptive fields will increase the overlap of the population of neurons that can potentially be activated by each presented stimulus, but the selectivity for particular types of stimuli, such as faces, can, when the other stimulus is not a face, reduce the overlap of the potentially activated population. So the prediction then is that maybe we will see evidence for multiplexing, and I'm going to describe exactly what I mean by that in a moment, and that we might see this more for two faces than for a face paired with an object that is not a face. So the data set that we received from the Frywald lab involves two kinds of stimulus combinations. In some cases, there's a face presented at one location. That we'll call that the preferred face. There can also be a non-preferred face presented at another location. And then the third possibility is that both of those stimuli are presented together. They also included trials that involved a face paired with some other object that's not a face. The analysis that I'm going to describe to you involves analyzing all three of these different basic trial types. First, the single stimulus trials help us establish what firing rate do we expect for each stimulus when presented alone. And then we will look at the firing rates observed on the combined dual stimulus trials to see if there's any evidence for fluctuating between the response level that is observed when stimulus A is presented alone and the firing rate when stimulus B is presented alone. So specifically, we're working with trial-wise spike counts. So we're looking for evidence of fluctuating activity across trials, not within a trial. And we did this because basically it worked. And so if you imagine when you present stimulus A alone and you get a Poisson distribution of spike counts like that, and you present stimulus B alone, you might get a different Poisson distribution of spike counts. We can then ask, selecting out conditions where the A responses and the B responses are Poisson and reasonably different from each other, and those are necessary conditions for our modeling. We can then ask, on the dual stimulus trials, what do the response patterns look like? We have three foils to this model, Bayesian model comparison. They all three involve various forms of single Poisson distributions of spike counts. Maybe the neurons seem to add up what they would otherwise have responded, how they would otherwise have responded, and then we'd get a single Poisson that's outside the range defined by the A and B responses. Or maybe there's winner-take-all for the best of the two stimuli. We'll call that a single. Or maybe there's averaging, in which case you'd get a single intermediate Poisson between the A and B response rates. That could also occur if there is fluctuating activity, but the fluctuations are occurring faster than the trial timescale. Nevertheless, our key model outcome of interest here is going to be whether or not we see a mixture of two Poisson distributions that are anchored to these benchmarks that we observe with the A alone and the B alone trials. So to just recap, these are basically our foils, and this is going to be the outcome of interest for what I'm going to tell you about. We got data from two different face patches. First, I'm going to show you data from MF. This is an intermediate in the face patch hierarchy. And we see in example cells evidence for this bimodal pattern of responses. Across the population, we see a distribution of different modeling winners, but mixtures are a prevalent category in this data set. When the faces are paired with a non-face object, we also see some mixture response patterns at the individual neuron level, but at the population level, the proportion of those is slightly lower than it is when the two stimuli are both faces. And that turns out to be a statistically significant result. In area AL, which is later in the face patch hierarchy, the picture is a little different. We see both mixtures for both face-face and face-object pairs. But the difference between these two is not significant. In fact, the mixture response pattern dominates the remaining categories to a greater degree than what we see in area MF. So to just pull up the data from MF on the same slide, you can see that there is a bigger difference between the mixture and non-mixture categories in area AL than in area MF. So to summarize this part, there is an interplay between specialization and this form of evidence for multiplexing. We do see more mixtures later in the hierarchy, but at a mid-range of the hierarchy, we see more mixtures for face-face than face-object pairs. This is broadly consistent with expectations from competition for representation. In other words, the degree of overlap in the potentially responsive population, and the parts that don't quite fit with this may, I think, be informative about our emerging understanding of the hierarchy of the face patch system. The last part that I want to tell you about comes down to objects. Light is continuous, but the physical causes behind those patterns of light are not. And our brains are capable of making inferences about where one object ends and another begins. When we talk about stimuli, what the brain is really trying to do is figuring out about objects. So now I'm going to turn to a data set, several data sets from area MT. And these involved either two distinct objects. In one data set, we have two gratings presented adjacent to one another. The monkey in this task is actually attending to a third grating outside the region of the receptive fields. A second data set comes from previously published work from Stefan Troya's group and involved two random dot patches also presented adjacent to each other. The monkey in this case is attending to a fixation point and detecting a change of that. A third data set is very different from the other two. It also involves gratings, but instead of presenting the gratings side by side, the gratings in the combined stimulus case were presented on top of each other, forming a fused object. So just once again to highlight where we got this data, the random dot is published data from Stefan Troya's group, and these two data sets also published from Marlene Cohen and Doug Ruff, now at the University of Chicago. What we see in these data sets is really very interesting. For both of the distinct object cases, we see mixture response patterns, both at the single unit level and at the population level. But we do not see that when the gratings are presented on top of one another and form a fused object. And this difference in the existence of mixtures in this MT datasets is quite significant. So just to summarize what we're arguing here, we're arguing that multiplexing may be a general strategy that's permitting preservation of information about multiple objects. This adds to our previous findings. We have performed similar analyses now in V1, V4, and in the auditory pathway in the inferior colliculus. Today's story, you can see the full version up on bioRxiv. And if this work is of interest to you and you're looking for a postdoc, we are recruiting. And you can view both the links to these papers and the job ad at that QR code. And finally, I'd like to thank our funders and acknowledge once again our collaborators and Meredith Schmale, who's first author on this work. Thank you.
Do we have any questions? Laura.
Hi, thanks for a really beautiful talk. So in what you presented, it was a two-dimensional problem. I just wanted to ask, how easy is it to scale this to a multi-dimensional scenario? And would that lead to kind of physiological limits that would kind of set up hypotheses in that regard?
You mean like three-dimensional, like including depth, for example, or?
Just like other features that are also kind of
right. We haven't actually done that, but we would be delighted if anybody has a data set that has that kind of A-B combo. We'd be more than happy to look at that. And I had an interesting conversation with someone who's been investigating occluded visual stimuli and the preservation of information about those stimuli.
Yeah, so a question about the time scale of the multiplexing. So you sort of imply that you are switching from one to the other representations, and I wonder if there's the characteristic time scale involved in that.
Yeah, so we have in some other work found evidence for faster switching as well, and those do tend to fall into that intermediate category here when we can identify them. These particular data sets, were, in some cases, the stimulus presentations were pretty short. And so if they're too short, it's hard to dig deeper to look for that kind of faster switching. Yeah.
Are you able to put sort of a time scale on it? Like, is it every 10 milliseconds, every 100 milliseconds? Or?
Well, these, I don't remember off the top of my head, but these are going to be 200 to 400 millisecond stimulus presentations. And so we're lumping it all together in that time scale. And then there will be a few hundred milliseconds before the next stimulus presentation at least. So we actually really don't know what kind of time scale we're talking about, but that should give you a order of magnitude of what we're able to uncover with this statistical method.
Okay, thanks.
Yeah.
I think we can just take one very quick question while Clementine is setting up.
Hello. Very nice talk. I was just wondering if you have any predictions of how you think attending one to one or the other object is going to affect the joint responses. Is it going to enhance only one of the responses, or?
As best my guess is right now, it does not. That this will occur, this occurs both when you're not attending, it occurs when the behavioral report task involves, you have to report both, and it occurs in an attention task where the animal is cued to attend to just one of the two stimuli. But we don't have, for that last argument, we don't yet have the ideal data set for making that claim.
Great, let's thank Jennifer again. Okay, we're just waiting for the Mac to stop being slow. It'll just be a second. Patience is a virtue. So I will take the opportunity here to remind everyone, I was asked to remind you all, if you're coming to the keynote tutorials tomorrow, which I hope you all are, a reminder to please charge your laptops beforehand, because there may not be access to power in the room. So, hope you've all noted that. Great, look, it made the talk come up. Okay, so next talk is Clementine Dominé from UCL.
So hi everyone, I'm Clementine Dominé, and for anyone wondering why there's an orange on the slide, it's because it's a clementine. Now we can get started. So let's imagine you and your collaborators have just recorded a new experimental data set, and you're thinking about modeling it or comparing it against existing theory or with a new model. There's a lot of steps that separate you from your end goal. First, you'll need to access, sort, and visualize your data. Then you need probably to create an environment in which your agent will be able to run. And most likely again, you need to run some hyperparameter search before you can move on to the result quantification and the result visualization. And this is a lot of work. And it gets worse. This work is, in part, or all of the path is often repeated to attempt to build a comparison between the different experiments and theories present in the literature. And in fact, a good comparison or standardized comparison would involve matching each experiment with each theory. But this is even more work. Scaled to the size of the experimental and theoretical literature of the hippocampal cortex, and in consideration of the expanding dataset sizes, it is really too much work. Altogether, it's really difficult to compare theories systematically against each other and against a full range of empirical data. To address this gap, we introduced NeuralPlayground, an open-source standardized framework that aims to standardize the comparison of the hippocampal and cortex models with existing literature. For example, upon the introduction of an experiment, it enables the automatic comparison of the model with the implemented theories. Other great packages, such as NeuroNav and RetinaBox, have been developed with broadly the same goal, but a slightly different focus, and I highly invite you to check them out as well. Concerning NeuralPlayground, the focus thus far has been on the hippocampal and cortex. The choice of this brain region was motivated by the presence of a large body of empirical and theoretical work. This brain region contains many types of cells, such as grid cells, head direction cells, and place cells. Together, they are thought to constitute the cognitive map that allow mammals to navigate efficiently and flexibly. So what is inside NeuralPlayground? How does it work? And how does it help with the standardization of the comparison? On the one hand, we have the agent class, which contains de novo implementation of three influential models of the hippocampal and cortex, namely the successor representation by Stackenfeld et al., the excitatory-inhibitory plasticity model by Weber et al., and the Tolman-Ackenbaum machine by Whittington and colleagues. These span a broad class of models, including RL, cognitive deep learning models, and system neuroscience circuits. This really shows the versatility of our framework. The second component of NeuralPlayground is the arena class. It provides the agent an environment in which it can explore and potentially learn over time. Any two-dimensional arena can be built using walls as construction units. This allows complex architecture to be implemented, such as the T-maze, cycles, and dynamical environments such as merging room to be implemented. The agent and the arena interact broadly like in OpenAI Gym. The agent receives observation from the environment, so for example, reward, visual cues, etc., and uses this to take action, which in turn will update its own state and the state of the environment. This aims to really mimic the interaction that the animal has with its environment. However, in addition, NeuralPlayground incorporates the experimental class. This is designed to centralize and standardize access to experimental data. The framework currently contains 3 influential datasets in the standardized label format, namely we have Hafting 2008, Sagolin 2006, and Wernhall 2018. Moreover, the data extracted from the experimental class can be used to construct arenas that will replicate the experimental setup. For example, the animal will be able to take action and take the exact same path as the animal in the arena. As a result, the agent will ignore its own actions and follow the path of the animal. A second noteworthy aspect of NeuralPlayground is the capability of the agent to iteratively update its internal neural representation as it navigates the environment. The plot rate map method facilitates the visualization of this representation. All right, let's look at an example to get a better understanding of how NeuralPlayground is functioning. So to begin, we initialize the experimental class Sagolini 2006 data, which contains information from Sagolini 2006 experiment. In this study, the researchers recorded neural activity from the entorhinal cortex of rats navigating in a square environment. The study revealed the presence of grid-like representation in the neural activity as shown here. Next, we utilize this information to create the arena. With the aid of NeuralPlayground, it becomes effortless to visualize both the recorded rate map and the trajectory of the animal in the arena. For the agent, we choose the SR model, developed by Stackenfeld and colleagues. After walking around in the environment and updating its model, the agent is able to create this type of grid-like representation. For comparison, we now repeat this process of update and plot with a second agent, the excitatory-inhibitory plasticity model developed by Weber and colleagues. Furthermore, you can also run the same agent on a different arena. For example, we now look at the Hafting 2008 experiment. In this study, the researchers recorded neural activity from the entorhinal cortex, navigating this time in a linear track. And the study reveals again the presence of grid-like representation in the neural activity. And we can now build the corresponding arena and let the agent train in it. Calling again the plot rate map function allows you to get a look at the rate map built from the Weber agent as it runs down this linear track. And for comparison, again, we also run the Stackenfeld agent in the Hafting arena. The agent is also able to build a rate map, shown here while navigating up and down this linear track. Thanks to the comparison board class, we can easily summarize and visualize our result. So far, we have run 2 different agents in 2 different arenas, namely the Sagolini arena and the Hafting arena. And this was done to be able to compare the neural representation of the models to the 2 corresponding experiments. But one can also run many more agents and many more experiments. Note that this process is actually automatized thanks to a simulation runner that allows you to pick any agent, experiment, or arena and run simulation automatically in the background. The output is then directly fed to the comparison board. To obtain a quantitative measure of the comparison, we also compute the gridness score as defined by Sagolini et al. 2006. This is allowed by the grid score class implemented in our framework. And you can actually also implement any other relevant score or matrix that you would like as a new class inside the framework. Note that we deviate from usual benchmark as we do not have a single summary number to measure the performance of our models. This is due to the diversity of ways experiments and models are done, and instead what we do is automatically generate several matrices and plots to quantitatively and qualitatively compare the results of models with experiments. In other words, the software can be viewed as a sandbox environment in which you can play around with agents and experiments, hence the name NeuralPlayground. Altogether, NeuralPlayground aims to facilitate comparison of experiments with models. But importantly, it does not specify it. We took the example of a grid-like representation, but multiple other representations can be investigated. For example, place cells or head direction cells, or any new representation your experiment might find and their corresponding models. The NeuralPlayground open source software was built to be collaborative and long-lasting. All contribution to the repository are acknowledged, and here is our beautiful collaboration so far. We welcome new collaborators to help us develop this framework. We have many new ideas for the future, but we welcome new ones. And this long-term community project is supported by the Gatsby SWC Neuroinformatics team. They are a team of professional software engineers that are dedicated to maintain the package. They will help us with maintaining libraries, implementing tests, introducing bots that will really help with the workflow. So if you don't trust me, you can trust them, definitely. Altogether, we hope that more researchers join our project, eventually leading the way to the computational understanding of the hippocampal and cortex. To start, to contribute, scan this QR code and check out our GitHub repository. You'll find there all of the information you need to get started and become a developer by adding a dataset, adding your model, comparison matrix, for example, creating a richer environment, or simply discuss the future of the framework with us. Or you can also join as a user by running comparison, exploring datasets, and playing with implemented models. I would like to thank the organizers, my collaborators, the Sax Lab and the Barry Lab for just being great and super supportive environments, and you, of course, for your attention.
Amazing. We have time for questions. People have any questions? Just a reminder that you can also post your questions on the dory. We have not had any dory questions yet for this session, as far as I know. Okay. I'm going to ask a question, which is, so it looks like absolutely amazing, the tool. I was thinking that perhaps, is it also built to be able to make predictions about, so if you're an experimenter who is planning an experiment, can you use the tool to make predictions about what you would expect to see under different model classes, either there or your own? Because you described it in terms of sort of post hoc modeling of your data. So is there a way to do that?
I think the other way around could work as well. You could pick some models that you think are suited for your experiment, create virtually your experimental setup, and see some of the predictions you could make for your experiment.
Yeah.
Definitely, it's very flexible.
So I guess you would need perhaps to have a model of what the rat would do. So maybe you could plug into rat in a box or something, and that would give you you could then use those trajectories, and then that would give you a full neural prediction of what you might expect.
Yeah, definitely. You can either have just any policy of navigating you could choose, but you could also plug in rat in a box, which is also one of our plans to get the naturalistic behavior paths. So yeah, definitely, this is definitely possible.
Fab. Any more questions? Okay, so let's thank Clementine again. Fantastic. And the next speaker is, I'm sorry, I'm going to say your last name wrong. Wiktoria Luczak. Okay, forgive me, I should have practiced that beforehand. And she's also from UCL. And the talk is, you probably need to press down and it will work. Cool.
Hello, everyone. Thanks so much for having me here. This is my first talk ever, so this is a huge thing for me. I'm currently starting my DPhil here at Oxford, and I've recently graduated from UCL, where my supervisor was Professor Steve Fleming. And we worked together on this project, which is my bachelor thesis, called Confidence is Detection-like in High-Dimensional Spaces. And this is what I'm going to present to you today. So my project belongs to the field of metacognition, which investigates how we reflect on our cognitive processes. And it's often quantified by judgments of confidence in perception, memories, or decisions. So good metacognitive abilities are associated with confidence accurately tracking changes in performance. However, certain cognitive biases may lead to systematic inaccuracies in metacognition, and one of them is the positive evidence bias that I'm going to talk about today. So positive evidence bias was initially found by Zilberberg and colleagues in 2012, and they used this classic random dot motion task paradigm, where participants watch chaotically moving dots, and they have to make a decision whether on average they move up or down, and then report their confidence. So what they found is that this choice, up or down, was predicted by both the amount of movement upward and downward with the same strength. However, the confidence reports were only predicted by the amount of movement supporting the chosen option. So it looks like as soon as participants made the choice, they started ignoring evidence opposing their decision when forming confidence. So one consequence of that is that it is possible to drive confidence up or down by manipulating the total amount of evidence, even when we keep a difficulty constant. So for instance, in this study from our lab, Rolwich and colleagues 2020 kept a signal-to-noise ratio stable, but increased or decreased the total amount of evidence. So they created a low positive evidence condition with a low signal and low noise, and high positive evidence condition with high signal and high noise. And what they found is that despite no difference in the accuracy, participants were significantly more confident in high positive evidence conditions. So it looks like we can drive somebody's confidence up by increasing all evidence, even if we don't increase their accuracy. And this again suggests that indeed confidence estimates are relatively insensitive to evidence against first-order decisions. And then quite recently, Webb and colleagues 2023 found the same effect using deep neural networks. So they basically took these digits from the MNIST dataset and they superimposed randomly scattered pixels and manipulated contrast. So they also ended up having high positive evidence and low positive evidence conditions. And then they trained neural networks to first classify the digit that was presented to them and then to estimate their confidence, so probability of being correct. And what they found is that similarly, despite no significant difference in accuracy, neural networks were significantly more confident in the high positive evidence condition. So why do we see positive evidence bias? Historically, it was thought to be just a suboptimal heuristic, so a natural consequence of human limited metacognition. However, the fact that neural networks that were specifically trained to estimate their confidence also show the same bias as humans suggests that there may be something more going on here. So Webb and colleagues propose that positive evidence bias might be caused by optimizing statistics of naturalistic sensory inputs, so a mismatch between experimenters and models' assumptions. And in this project, we further investigate this idea that PEB might be a mismatch might come from a mismatch between experimenters and participants' assumptions. So we hypothesize that it might occur when participants compute their confidence in higher dimensional space than that assumed by experimenter. So what do I mean by that? For example, we can imagine experimenter asking question, is it left or is it right? But subject actually accumulating evidence for left or right or something else. So they're in higher dimensional space. So in order to understand metacognitive failures, we need to investigate computational basis of confidence formation. So here we treat confidence as a base optimal probability of being correct given the evidence, and we use this simple signal detection theory model, and we look at discrimination and detection tasks. So this is from a paper that is currently published from Mazer and colleagues 2023. So in this discrimination task, we can imagine choosing between S1 and S2 and gathering evidence on the respective axis for them. So if we're in this purple area, we should always choose S1, and on the green area, we should always choose S2. Now, on this white diagonal line, we should not be able to choose, as we have equal amount of evidence for both the options. But the further we deviate from this line, the more confident we should be that our choice is correct. So our confidence should be driven mostly by the relative evidence, so how far we are from here. In contrast, in detection task, we want to know if we detected something versus nothing. So we want to be as far as possible, so we're summing evidence, so we want to be as far as possible from the origin of the plot. So this confidence that we have in this task, so the specific quadrant that we're on here, will be mostly driven by the amount of evidence for the dominating option. Because we can imagine having a lot of evidence for S2, little evidence for S1, and then increasing evidence for S1 will not really influence on our confidence anymore, but evidence for S2 will. So if you may remember that this is very similar to what happens in positive evidence bias, where participants form their confidence depending on the amount of evidence for the dominating option. And further in this talk, I'm going to explore what happens to these confidence criteria when we increase dimensionality of the space. So how do we do that? We start from bivariate Gaussian model and we extend it to higher dimensions. So each new stimulus will be new orthogonal dimension, and we can consider optimal confidence function based on specific dimensionality. So we fix dimensionality k, that must be at least 2. And for each stimulus from 1 to k, we assume that they follow this xi k-dimensional Gaussian distribution around the mean of Si, where Si is a vector with 1 on the 8th coordinate and zeros elsewhere, and sigma is a variance-covariance matrix that we assume to be an identity matrix. So we basically assume that all our stimuli are equidistant from the origin, they have the same variance, and all the dimensions are independent. And then we calculate confidence as a Bayesian posterior probability of having this option giving evidence that we see. And this is how it looks like. So just to explain what's happening here, for 2 dimensions, we may be choosing between, let's say, 0 or 1, so we have 2 options. In 5 dimensions, we may be in an environment of having 5 options, like all even digits. In 10 dimensions, we may have all digits, and we can also imagine having higher dimensional spaces. So we obtain these 2-dimensional plots by, for each dimensionality, we fix non-evaluated stimuli at low levels, and we're taking 2-dimensional slice through the two options that we are considering. And we can compare a choice between x1 and x2 depending on how many other dimensions are available. And we can see that as we increase dimensionality, it begins to look detection-like. So the effect is stronger as we have more dimensions. And this is just to clarify the resemblance of a high-dimensional discrimination surface. So this discrimination looks like this. And when we increase dimensionality, it begins to look detection-like. So as you may remember, positive evidence bias is congruent with this detection-like function. So what we thought to be a suboptimal bias in discrimination confidence may in fact be explained by subjects computing optimal confidence in higher dimensional space. So this is how we can illustrate the positive evidence bias by these plots. So let's assume that we are in 10-dimensional space, but we're choosing between x1 and x2. So we can fix all the 8 non-evaluated options at a relatively low level. So let's say 1 plus minus epsilon, and we fix x2, that is available option, as 1.5. And then we can see what happens to how the posterior and confidence changes based on the amount of evidence for x1. So we can see that as we increase evidence as we increase x1 until it reaches the point of 1.5, so it equalizes the x2, we should choose x1. And then as soon as it crosses this threshold, we should choose x2, sorry. And as soon as we cross this threshold of 1.5, we should start choosing x1, because this is now the dominating option. And then when we look at confidence, depending on x1, we can see that when x1 is not chosen, it has very weak influence on confidence. So it's very much compressed by, suppressed by other stimuli. But as soon as it crosses this threshold to be chosen, it starts to have very strong influence on confidence. So we can see it here, that when we move here, confidence doesn't change, but as soon as we cross this threshold, we have quick increase in confidence. So we basically derived it from the Bayes theorem. So we assume a Gaussian likelihood and flat prior for all the digits, and then for two-dimensional discrimination, we will end up having this function that is linear, and it depends on the ratio of likelihoods for the two evidence that we're considering. But as we add more stimuli, so we increase dimensionality, we have to normalize by the higher number of components, and this is what induces this nonlinearity in the function. So then we decided to test it empirically on neural networks. So for the same perceptual task, we decided to change dimensionality of the set to see what happens as we manipulate dimensionality. So you can imagine the same task, so recognizing digits like here. So you should say that this is 2, but sometimes you can be in two-dimensional space when you're wondering, is it 2 or is it 4? And another time you might be in 5-dimensional space, so you're wondering which of, let's say, all even digit this is. So we expect that being in higher dimensional space, even if you have the same task, creates this positive evidence bias. So we modified the original Webb and colleagues algorithm, so neural networks were trained on different number of classes, so between 2 and 10 dimensions, and for each dimensionality we chose digits randomly. And we want to check whether PEB increases with dimensionality, so we expect to see an interaction between positive evidence level and dimensionality in their influence on confidence. So we already know from the Webb and colleagues paper that for 10 dimensions, for 10 digits, there is a strong positive evidence bias. So here we're asking, if we decrease number of classes, if the effect that we see will become weaker and weaker and weaker. And this is what we see. So as predicted, despite no significant difference in accuracy between both conditions across all the dimensions, in each dimensionality, neural networks were significantly more confident in the high positive evidence condition, but this difference, so the positive evidence bias magnitude, was increasing as we were increasing dimensionality. So just to summarize, in the positive evidence bias, second-order judgments are insensitive to opposing evidence, and this used to be attributed to metacognitive limitations, but recent evidence suggests that it might be a computationally optimal behavior, like optimal adaptation to naturalistic stimuli. And here we show that Bayesian computations of confidence in high-dimensional space also demonstrate positive evidence bias. And we tested it empirically on neural networks, which showed that increasing dimensionality indeed increased the positive evidence bias. So in conclusion, we may say that positive evidence bias may be rational behavior if participants compute confidence in space that is larger than that assumed by experimenter. Thank you.
That was a fabulous talk. Do we have questions? Yes, Megan.
Hi. Thank you so much for the really lovely talk. Yeah, this is great stuff. So I had a question about the implementation of your Bayesian model or your signal detection theoretic kind of model. So you posit that each of the sources has an equivalent distance from the origin, and then you're doing the inference in that space. But it strikes me that the confidence contours that you're showing in higher and higher dimensions also look an awful lot like the shape of the confidence contours if you simply marginalize over all possible stimulus strengths, even in a two-dimensional version of this space. So I'm wondering how specific the dimensionality effect is to your choice of this kind of prototype-like model versus marginalizing over all possible stimulus strengths, even in just a two-dimensional STT regime.
So basically, yeah, so this model, I think, yeah, so the assumptions that we created for this model are quite simplistic, so we can imagine having different model that we base it on. I'm talking about this one, yeah?
Yeah, this one, or even if you go back 2 slides, or to the one that you were showing just before, I think.
This or?
The one with all the, yeah, that one. So the one with k equals 20 looks, like, those contours look like if you just do a marginalization over all possible x1 strengths and all possible x2 strengths, they look very similar in the contour space.
Yeah, so I mean, there are more things that influence shape of these plots. So one of them is dimensionality, but as shown here, basically the shape of this and this nonlinearity is induced by what's happening here. For example, so one thing is that we assume Gaussian likelihood distribution, and we assume that there's the same variance. So that's one thing, that if you can change this and it will look differently. And it also depends on the actual value of posterior for specific stimuli. So if you choose to fix your evidence on different level for different stimuli, it will also look differently. Yeah, so for example, you can imagine having all the stimuli as 0, or likelihood for all the other non-evaluated choices as 0, and then having a high evidence for the 2 dominating options, and then it will basically look linear, so it will be the same as this. So even in higher dimensionality space, it will be still linear.
Okay, we can talk more.
That sounds like a starting point for a longer conversation. Okay, one more question.
Hi, a great talk, interesting concept. My question is, you said that accuracy remained the same, but the confidence increased. But in case of neural networks and other discriminatory phenomena, you don't have a time limit in choice. If you impose like a time limit in distribution models, like fast decision times, accuracy changes because there's a speed accuracy trade-off, and that confidence come up as you increase your evidence. Higher confidence will result in faster decision time. So how do you comment on that?
Oh yeah, I think that's a really interesting point. I mean, in our empirical testing, we didn't use humans in this. There is no human evidence in this presentation, so we only use neural networks. But I agree. I think that there are even more differences in how humans would make decisions in this case. So these slides that I presented are not our studies; they are from other studies. But I agree that there are many other things that can be considered when we talk about how people make decisions. And yeah, and I think that it makes it difficult to investigate, but I think this is something that we will look into. Yeah.
Thank you.
Amazing.
And if anybody has any more questions and would like to maybe talk to me or Steve, then we will be waiting during the coffee break.
Fantastic. Thank you so much. What an amazing talk. Okay. And our next speaker is Mufeng Tang from here at the University of Oxford.
I guess it's good to be presenting here instead of taking examinations. Sorry, Victoria, we can't find the slides.
So it should be.
Oh yeah.
Is this yours?
Yeah, it's mine. I saw it's turned off.
Okay.
Sorry. Are we good? Yeah, we're good. Okay, thanks everyone for being here, and also I want to thank the CCN organizers for giving us this opportunity to present our recent work in predictive coding models for temporal prediction, which is a joint work by me and my supervisor Rafael, who's sitting over there, and also Baron and Mayar, who couldn't make it. So the motivation behind this exploration is that as humans or as animals, we make predictions, and in particular temporal predictions every day. For example, if you play basketball, you always have to predict the future position of the basketball when you're fighting for a rebound. And we know that in the brain, there are temporal predictive activities almost everywhere, for example, in the visual cortex and in the hippocampus. So the question we want to answer in this work is that how does the brain perform this kind of temporal prediction, and can we model these temporal predictions in a biologically plausible way? So a very natural and very good candidate for answering this question is predictive coding networks, because of course they naturally involve these predictive computations, and they also, in their network implementation, they require only local computations, making them biologically plausible. So the original predictive coding networks, which was proposed in this seminal work by Groh and Barnard in 1999, proposes that the cortex, it follows this hierarchical structure, where the higher layers will generate predictions of the activities in the lower layers, all the way down to the current sensory input, for example, a car. So if we abstract out the kind of probabilistic structure of this hierarchical model, we can think of it as this predictive model where the brain is trying to generate the predictions of the current sensory input using a set of parameters C. So in this model, the brain, which we denote as X, is the latent variable, and Y is the observable. So the objective of this generative probabilistic model is to infer the most likely brain activities X given the current observation Y, which if we skip all the Bayesian steps, we can, this objective can boil down to this minimization problem of this prediction error, squared prediction error, and this prior or the constraint on the brain activity X. So the first term is the prediction error, where we model the prediction as a nonlinear function consisting of a nonlinearity F and this matrix C. So the question now is, what if the car that we observe moves? So of course, the original Groh and Barnard static predictive coding model did not take into account the time, the temporal dimension. So in this work, what we would like to propose is a temporal extension of the original predictive coding network. So in this model, the hidden activities, latent activities X, do not only generate predictions of its current corresponding observations Y, but they also generate this temporal predictions of their own activities in the future. For example, in this probabilistic structure, you can see that xt-1 do not only generate predictions of yt-1 via C, but it also generate predictions of xt via the matrix A. So the objective function that we can derive from this probabilistic model is very similar to the static predictive coding. So the first term you can see that is actually exactly the same, which is the hierarchical prediction error squared. The only difference is that the second term now has a dependence. The second prediction error now has a dependence on the prediction from the previous time step, which we again model as a nonlinear function consisting of a nonlinearity F and this matrix A. So this model is what we call the temporal predictive coding, TPC. So now that we know the underlying probabilistic structure of it, we now talk about these computations and network implementation of the TPC model. So we can first define these two arrow values. One is epsilon x, which is the temporal prediction error. And we also define epsilon y as a hierarchical prediction error. And then we can also define this objective function as f, which is basically the sum of the squared of these two levels of prediction errors. So in this model, we can infer the hidden activities x following gradient descent. And if we just do all the math, this will be the expression that we will get. And we will also update the parameters, which are the weight parameters A and C, using gradient descent as well. And if we skip all the math, those are the expressions that we will get. So those expressions themselves are not important. What's important is that we can implement all these computations in a biologically plausible neural circuit requiring only local computations and Hebbian plasticity. So in this neural implementation, we introduce those arrow neurons, so in the observation level, it will receive excitatory input from the value neurons, which will just represent the observation input, as well as those inhibitory input from the higher level, which generates a prediction using the matrix C. And following the same kind of idea, we can also introduce those hidden arrow neurons, which are the, which are the epsilon x, which will receive excitatory input from its corresponding value neurons in the hidden layer, as well as those temporal predictions from this special set of value neurons, which we call x hat t minus 1. So similarly, the inferential dynamics, which is this xt dot, can also be implemented in this neural circuit using those excitatory inhibitory input, as well as the arrow neurons. And what's more important is that the learning rules for A and C are totally Hebbian. So if you take a look, there will just be the outer product of the presynaptic and postsynaptic activities. So I know this slide is a bit dense in terms of information. So I think the take-home message from this slide is that all these computations of the temporal predictive coding model can be implemented in this neural circuit requiring only local informations and Hebbian plasticity. So that is a model. So the first kind of result that we obtained with this model is that this temporal predictive coding model can approximate this well-known filtering algorithm, Kalman filter. So analytically, we found that both temporal predictive coding and Kalman filter are instances of this recursive Bayesian filtering algorithm, which essentially is just to infer the current posterior, which is p xt given y1 to all the way to yt, based on the previously inferred posterior, which is p xt minus 1, given y1 to yt minus 1. The only difference is that, so both of them do this, but the only difference is that the Kalman filter will assume when it's trying to infer the current posterior, it will assume that the previously inferred posterior is a Gaussian distribution, which is centered at xt minus 1 hat, but with the uncertainty or with a covariance matrix of sigma t minus 1. And it will try to kind of estimate both the mean and covariance of this Gaussian. Whereas our temporal predictive coding does something slightly different in that it does not estimate the sigma t minus 1. So it will assume that the previously inferred posterior is a delta function centered at x hat t minus 1. There is no uncertainty, no covariance. So in short, the temporal predictive coding will approximate Kalman filter when sigma t minus 1 goes to 0. I know you probably now have this question, will this approximation affect the performance of our temporal predictive coding compared with Kalman filter? So we set up this very simple simulation where we have these two latent variables xt, which are three-dimensional vectors, which are also transitioned with this matrix A representing Newtonian motion. So at each point in time, xt is a three-dimensional vector representing position, velocity, and acceleration. And we also introduce this random matrix C to totally scramble the Y so that you can no longer decode, well, visually probably, by eyeballing it, you can never decode what is the underlying X given the observations Y. So on the left is the true X, and on the right is the kind of scrambled and very noisy Y we produce from those true X. So the question is, can the temporal predictive coding model decipher the true states from these noisy YTs? So what we found that the temporal predictive coding, together with Kalman filter, can provide very good estimations of the underlying state. So what I'm showing in this figure is just the third dimension, which is the acceleration, but it's the same performance on the position and velocity. So we found that both Kalman filter and temporal predictive coding will perform quite well in this task, and more importantly, the temporal predictive coding will approximate the performance of Kalman filter in this task. So you can see that these two curves overlap very closely. But one important thing I want to note is that temporal predictive coding achieves this using a neural circuit I just showed with only local computations, whereas if you are familiar with Kalman filter will require a lot of complex matrix operations like matrix inversion. So another question you're probably going to have is, uncertainty is good. Uncertainty is good in both artificial neural networks and in the brain. So is there a way that the predictive coding models can keep estimating it? We did not address this question in this work, but I would like to introduce my colleague Gaspar's work, who is sitting among us probably, but I couldn't find where he is. So so what he found was that predictive coding can, in some way, represent this uncertainty. So if you're interested, you can go catch him after this presentation. Okay, so that is the analytical or theoretical result we found with temporal predictive coding and its connection to common filter. And we also tested temporal predictive coding in a lot of neuroscience-related tasks. One is this perceptual aliencing task. The challenging bit of this task, so if you look at this example on the left, the challenging bit of this task is that if you look at this sequence of stimuli where a white bar is first moving down and then up, the second frame and the fourth frame are exactly the same. So if you want to, based on your current observation, which is on the second or on the fourth frame, and trying to make a prediction of the next frame, you'll have a problem because they are exactly the same. So what we found, so this is called the perceptual aliencing problem. So what we found is that this temporal predictive coding can solve this problem and predict the next frame quite well after the second and fourth frames by developing different latent representations of them. So in this figure, the top row is the latent activities of the value neurons in a temporal predictive coding model, where we simulated 5 neurons because those are quite small stimuli. And we found that the kind of latent representations of these two identical observations are quite different. That's how the temporal predictive coding is indexing the kind of contextual information and making the correct prediction of the next time step. So we also tested temporal predictive coding in memory and recall of sequences. We first simulated this sequence of random MNIST digits and asked the temporal predictive coding to memorize it. And during recall, we provide a cue, which is the first image, and we found that temporal predictive coding can recall the full sequence, and also tested in this clip from BBC in 2012 Olympic Games. And we found these temporal PC can also memorize and recall these even high-dimensional videos, movie clips quite well. And now we're talking about memory. Another step, we took one step further and did some preliminary explorations into the generalization capability of temporal predictive coding. So during the training phase, we presented to this TPC with 5,000 rotating MNIST. So each row on the left is one sample, which is the MNIST digits that's rotating. And during testing, we provided with this model a cue, which is an unseen image that we sampled randomly from the eMNIST dataset. So we found that this model can well generalize this rotational structure to this unseen digit and rotating successfully this unseen character. So that is everything. So in summary, we introduced this temporal predictive coding model, which affords a plausible neural implementation with local computations and Hebbian plasticity. And we also show that there's an analytical link between temporal predictive coding and common filter. And we also found that temporal PC can work well in a lot of cognitive tasks, such as de-aliencing, memory, and generalization. And thank you very much. And if you're interested in the preprint, you can scan this QR code here.
Fantastic. Questions?
For the great talk. I was curious, given that you get into the implementational stuff, do we have any evidence that we have error neurons also in the brain?
There are some evidence of error neurons in the brain, but the problem is that's like a long criticized property of predictive coding networks. But I think the thing is not about whether error neurons exist. The problem is whether they have this one-to-one connections with their corresponding value neurons. That's something that's a little bit implausible in these models. But to answer your question, error neurons, there are evidence for that. Yeah.
More questions? I have a sort of... Oh, yeah, go for it.
So you showed in the end how the rotation is very easily captured. But how do you deal with like nonlinear, like not matrix capturable kind of transformations?
That's something we're actively investigating, because those are just preliminary results in generalization. Yeah.
Thank you.
Thanks for that suggestion.
Okay, looks like a really promising model. Fantastic. Okay, we're just going to set up the next speaker, who is Fenying Zang from, I forgot,
Leiden.
From Leiden University. I knew that. Fantastic. Over to you, Fenying.
Thank you. Hi everyone, I'm Fenying Zang, a first-year PhD student from Leiden University. So it's really a great honor for me to be here to introduce our project, Age-Related Changes in Neural Variability in the Decision-Making Task. Age-related changes are reflected at different levels. From psychological perspective, aging is characterized by impairments in decision-making, memory, learning, and many other cognitive functions. Also, biological research found that aging causes cellular neurodegeneration. But how does biology really change cognition? One potential way is those classical theories from last century. They state the neural variability increases with age, and thus the effective signal-to-noise ratio in the central nervous system decreases with age, which may finally lead to the cognitive decline. However, limited by the technology, those theories were not tested directly at that time. Recent research on anesthetized monkeys found that both single neural variability and noise correlations increase with age. This result is consistent with the first part of those classical theories. But since they used anesthetized monkeys, so they can't link the neural changes to behavior. So in our project, we want to explore neural variability in large-scale neural recordings from behaving mice. In this way, we can ultimately link the neural changes to behavior. Also, by using mouse model, we can include more individuals in our analysis. We also want to quantify the neural variability in other neural structures beyond cortex, because those previous research, they only focused on the cortex. We don't know what's happening in other neural structures inside the brain. Our hypothesis is the older mice exhibit higher levels of single neural variability. This is our first hypothesis. It's also the basis of the whole project. If we can test this hypothesis, then we can really look into this kind of change induced by aging and link it to behavior. We used neuropixel probes to record the extracellular signals from those 5 brain regions simultaneously. As we can see from this image, the blank line, it indicates the targeted trajectory of our probe. It goes through from the posterior parietal cortex, PPC, to hippocampus, CA1, and dentate gyrus. Finally, it reaches the lateral posterior nucleus of thalamus and posterior nucleus of thalamus. Here, the right panel, it shows the actual trajectories of our probes. The neural signals were recorded during a visual decision-making task. Those mice were requested to judge the location of the visual stimulus on the screen, either on the left or on the right. And our dataset consists of 2 parts. The young mice data is a public dataset from International Brain Lab, IBL. IBL is a huge team science effort which replicated the same neural recordings in many neuroscience labs around the world. And the old mice data was recorded by my supervisor as a member of IBL. Both of them were recorded based on the same standardized protocols developed by the IBL. Then we will take one brain region as an example to illustrate the data analysis.
We used the fun factor to capture the single neural variability in the current analysis. The fun factor is defined as the spike count variance over trials divided by the spike count mean. So in our dataset, after quality control process, we first aligned all of the correct trials to the stimulus onset. Then, by using a sliding window method, the firing rate and the fun factor time course were calculated for each neuron under each condition, then averaged within this brain region LP. Here, those two lines are from the young group. Similarly, we got result from the old group. From the overall pattern, we can see the old group showed a smaller average firing rate and a larger fun factor or the neural variability than the young group. Then, if we choose two time points in the time course, one is pre-stimulus and another one is post-stimulus, then plot the distribution of fun factor and those two time points separately. The statistical comparison between groups indicate the old mice have larger post-stimulus neural variability than the younger mice. Also, there is another interesting phenomenon. As you may have already noticed, in the fun factor time course, there is a clear decrease here around the stimulus onset. Actually, this phenomenon is called the variability quenching, which means the variability of the neural responses decreased around or on the stimulus onset. Here, similarly, the fun factor decreased. And this phenomenon has been widely observed in various cortical regions and species. Here, we also observed it in the internal neurostructure, LP, beyond cortex. Then similarly, if we use those two time points and calculate the variability quenching or fun factor quenching for each group, and make comparison between groups, the result indicates the old mice showed smaller variability quenching than young mice in the current brain region LP. In the previous slides, we were focusing on the LP brain region, lateral posterior nucleus. Now let's move on to look at other brain regions as well. Actually, the posterior nucleus of thalamus and the dentate gyrus of hippocampus, they showed the same pattern or the similar pattern as LP, which means the old group showed a larger neural variability than the young group. But we observed an opposite pattern in the C1 brain region of hippocampus. And we didn't find a significant group difference in the PPC brain region. Actually, the result of PPC brain region is kind of surprising to us because it's inconsistent with previous research on monkeys. We're still exploring the reasons. Then, if we focus on the variability quenching or fun factor quenching, we found all the 4 brain regions, they showed the similar pattern, which means the old group showed a smaller variability quenching than the young group. Again, we didn't find a significant group difference in the PPC brain region. And to summarize, in our project, we examined the effect of aging on the neural variability by using large-scale extracellular recordings from mice. And we indeed found the aging effects. As for neural variability, we found the old animals showed higher trial-to-trial neural variability in thalamus regions and dentate gyrus of hippocampus. As for variability quenching, we found the old animals showed smaller variability quenching in thalamus and hippocampus. And we have to mention the aging effects are not entirely consistent across different brain regions we have examined. And in our following analysis, we want to look into different cell types. We will also calculate the noise correlations between neurons to extend our current analysis from the single neural level to the local circuit level. Finally, we will link the neural changes to behavior to look into the behavioral consequences of this kind of change. Ultimately, we want to understand aging from a mechanistic perspective and understand how aging changes neural computation. In the end, I want to say thanks to our co-authors, especially IBL. If you are interested in the public dataset we're using, you can visit IBL website for more information. And special thanks to my supervisor, Anna Ulai, and our lab. And if you have further suggestion or feedback to our project, please come to our post and discuss with us. Thank you.
Fabulous. Well done. We have, sorry, I don't know who was first. Maybe we'll start with this one here.
Wondering why you are using the Fano factor as a variability measure. In your introductory slide, after all, you use the signal-to-noise ratio, which seems more relevant for behavior. I mean, if you have a Poisson train that goes from, let's say, 100 hertz versus 10 hertz, the Fano factor will be the same, but the signal-to-noise ratio will be much higher for the high rate, and that seems to be the thing that's relevant for behavior, I would think. So why the Fano factor and not the signal-to-noise ratio? Or do you see the same things in it?
Actually, we just started our project, so we started from the Fano factor since it has been widely used in previous research to capture the neural variability of single neurons. So we started from Fano factor, but maybe in the following steps, we will also consider other measures as well, and also maybe compare them between each other.
Okay, thank you.
Thank you.
Hey, thank you for a very interesting talk. So you started with all theories that thought about variability as noise. In recent years, we stopped thinking about variability as noise per se. We know that variability is important for learning and so on. Were you considering variability here as a compensation mechanism, for example, for the neurodegeneration, especially as you're going to link it to behavior later, but even just to understand what you see there, is variability there noise or is variability signal?
Actually, according to my understanding, yes, in the decades ago, the previous research, they think the neural signal or neural variability is kind of a nuisance. But recent years, the new researchers, the new findings found the neural variability plays an important role during the information processing or the encoding process. So we started our project. We want to try to explore if it's really play an important role in this kind of process. And in our following analysis, we will link the neural variability to the decision-making behavior, and as I just mentioned, and we will maybe use the computational modeling process to maybe drift differential model to capture the features of the decision-making process and link them to our neural variability. Then maybe we can answer your question.
Thank you.
Thank you.
Could I just sneak a follow-up question? I don't think you said whether the older mice the behavior was different to the younger mice. Were they worse on the task than the younger mice?
Actually, in current analysis, we didn't look into the behavioral part. Yeah, maybe.
So maybe that would help answer the question. Sorry to steal your thunder.
Thanks for the great talk. I was curious, the older mice did the same task before, like the younger mice, or like they are just another population of mice that they are basically just controlled?
Oh, sorry, I didn't.
So maybe another form. So the older mice are more practice mice in your experiment?
No, yeah, they're the same.
Super. Okay, let's thank Fenying again. Okay, and our final speaker of the session is Peter Murphy, who's at Maynooth University now, I think. And he's going to talk about information integration.
Hi everybody. Thanks to the conference organizers and the reviewers of our paper for selecting us for this contributed talk. I'm going to tell you about work that my collaborators and I have been doing to interrogate a possible shared neural circuit for the maintenance and integration of information over time, two computations that are central to working memory and decision-making, respectively. So I'm going to start just by quickly linking in with another session from the conference today, which was the generative adversarial collaboration on reconciling Sherringtonian and Hopfieldian views on neural computation. So it was alluded to during the session and made more explicit in a really nice perspective piece recently from Tatjana Engel and colleagues that a recent line of research has culminated in what I think at least is quite an instructive convergence between these two views. So this work establishes pretty explicit links between single-cell tuning curves and circuit structure, dynamics on the neural manifold, and ultimately behavior. And specifically, this is work aimed at understanding the head direction system of the fly. It suggests that this system can be understood as a so-called ring attractor, where neurons with tuning curves that tile the space over which the task variable, in this case, heading direction, varies. These are endowed with excitatory and inhibitory connectivities that conspire to produce a self-sustaining bump of activity over the population. This bump represents the animal's estimate of their current heading direction, and it also moves around a low-dimensional manifold, in the case of a classic ring attractor, a one-dimensional manifold, as this estimate of heading direction changes. So this work is exciting, I think, partly because it builds a bridge between these two dominant perspectives on neural computation. And indeed, other research, though maybe not yet quite as conclusively, has established similar links between attractor-type circuit motifs and other cognitive functions. So the work that I will tell you about in my talk focuses on these latter two functions highlighted here, working memory and decision-making, and it attempts really to address the question of whether we see signatures of an attractor-like circuit in action as human participants perform working memory and decision-making tasks. So one idea that has emerged from work in these areas is that a single neural circuit can, in principle, perform both of these functions. So it can maintain information stably over time in the service of robust working memory. In the case of the classical ring attractor, this is achieved through generation of a bump of activity over the population which persists in approximately the same location during working memory delays, even in the presence of distracting information. And if tuned appropriately, this same circuit should be capable of flexibly integrating multiple pieces of information over time, which is the key computation for deliberative decision making. So in a ring attractor, this would be achieved through the population activity bump moving around the ring manifold in a way that tracks, for example, the perfect integral of all information encountered on a trial of a decision-making task. So it's this claim really of shared circuits for both maintenance and integration of information that we hoped to try to put to the test with our work. So we've been taking a few different approaches to do this, and these have all involved interrogating the same set of cognitive tasks. One of these is a spatial working memory task in which a single target stimulus is presented anywhere along a circle. Participants need to remember the spatial location of this target over a subsequent delay period, during which a distractor stimulus offset in time and location may or may not be presented, and after which they need to reproduce the target location with a directed mouse click. So this task is very close to the classic memory-guided saccade task from the working memory literature. We also developed an integration task in which participants were presented with between 2 and 5 of such stimuli in a sequence, and their task here was to report the circular average of all stimulus locations once the sequence ended. And participants performed a simple sensory motor responding task that shares sensory and motor characteristics with the other tasks, but lacks any requirement for working memory or integration. And just to emphasize, these are tasks that require maintenance or estimation of a continuous circular variable, so they're well suited to being solved by a ring attractor. So we first set out to establish, I suppose, a kind of a proof of principle for the shared circuit model of these tasks by training recurrent neural networks to perform them and then interrogating the resulting solutions. So here, each network was trained to perform all tasks with sustained task rule inputs telling the network which task it should perform on each trial. In this example, a network is being instructed to perform the working memory task, and accordingly, it responds with the location of the first stimulus it encounters, and it ignores an distractor that's presented during the delay period. So in general, we found that the RNNs learned to perform the tasks very well. For a trained network shown here, recall error on the working memory task was low, and distractors exerted minimal, if any, influence. While on the integration task, the estimation error was also low, and appropriately for this context, analysis of the network's psychophysical kernel showed that it now gave approximately equal weight to all stimulus locations encountered on a trial, as required if it's integrating all information to estimate the true average. So this network, at least, was able to adapt appropriately to the two different task contexts. Importantly, the trained network seemed to produce this behavior by converging on the same shared circuit solution to the tasks suggested by the handcrafted attractor models. So specifically, in the trained networks, there was preferential excitatory connectivity between recurrent units that share similar location selectivity, and also net inhibitory connectivity between those with different selectivity. So this is really a central feature of the handcrafted circuit models. Moreover, many units showed strong persistent activity during the delay period of the working memory task that was selective for the content of working memory. So when arranging these circuits in a ring according to their preferred locations, we see the expected bump of activity form at the target location, which is marked here by the yellow vector. And this bump persists at the same location for the entire delay, despite presentation of a distractor indicated by the red vector partway through. On the integration task, however, the activity of the same units forms a bump that now moves around the ring to track the running average location. So here, the location of the running average is now indicated by the yellow vector, and the locations of individual stimuli by the white vectors. So what I'm showing here is a kind of a qualitative illustration that the trained networks appeared to be doing what the shared circuit model predicts. That is, the same population of units is representing both the remembered location during the working memory task and also the decision variable, so this evolving estimate of the average location during the integration task. We can also show the same thing more formally and quantitatively by decoding working memory targets and decision variables from the multivariate patterns of activity in the network. So applying this approach first to the working memory task, we find that the target location can be decoded very well from activity during the delay period, and that the underlying code generalizes strongly over time. So this is really just a complementary way of showing that this bump of activity that forms in the network persists in time, as was captured in the video on the previous slide. Importantly, though, there's also strong generalization of this code across tasks now to predict the evolving decision variable on the integration task. So in the analysis shown here, we trained a decoder to predict working memory targets based on unit activations during the working memory task delay period, and we then applied that same decoder to decode the evolving decision variable during the integration task, and we found that this worked very well. The relative decoding patterns of decision variables at different sequence positions, these are a little nuanced, and I unfortunately don't have time to go into the details on this, other than to say that it turns out these patterns that I'm showing here, they're entirely consistent with continued representation of the evolving decision variable given its autocorrelation profile. So this cross-task generalization of the neural code, it fits really with this central hypothesis from the shared circuit model that both memories and decision variables are represented in the same way in trained networks by the same population of units. And so really the take home from this for us was that this exercise with the RNNs, it provides this proof of principle that the shared circuit solution is a viable one for these tasks. We then look to assess whether this same solution is employed by the human brain by decoding memories and decision variables from scalp EEG data measured while human participants performed the same tasks. So here again are the findings from our decoding approach applied to the trained RNNs, which can serve, I suppose, as predictions for what we might expect to see in our neural data. And indeed, we found broadly similar signatures, so both in terms of temporal generalization of the code during working memory delays, and also a generalization of that code across tasks to represent evolving decision variables on the integration task. So it is worth noting here that there are some deviations between RNN and EEG. In particular, time-dependent decay in EEG decoding accuracy across both tasks that is not there in the RNNs. This could have several origins, including, for example, maybe a multiplexing of sensory and mnemonic codes in whole scalp EEG signals, as well as maybe patterns of behavioral error in human integration task performance, suggesting that we have less accurate estimates of internal decision variables, particularly later on in the sequence, and this is going to translate into worse decoding accuracy. But in any case, the key predictions of a neural code that generalizes both over time during memory maintenance and from memory to integration contexts, these are supported in these data. We can also ask, is there any evidence of the shared circuit in action in the behavior of our participants? So one prediction of the shared circuit model is that there should be a common source of noise in behavioral reports across both contexts. And this is going to be caused by random drift in the population activity bump over time. We do see evidence for such a common noise source and a correlation across participants in the magnitude of the behavioral error in both tasks. Importantly, this correlation is also preserved when covariating for error on the immediate sensory motor response version of our task, which is going to account for contributions of sensory and motor noise sources that are shared across all of our tasks by design. Another possibility is that working memory and decision-making behavior will be susceptible to the same forms of bias, if that bias indeed originates within the shared circuit. So one well-known bias in both literatures is a so-called history bias. This often manifests as an attraction of the current memory reports or decisions toward those formed on preceding trials, particularly if they're close to each other in feature space. So in our data, we found that this form of history bias was indeed present in both the working memory and integration contexts. Importantly, it was also completely absent in the immediate sensory motor response tasks. It's a bias that really seems to originate within the putatively shared memory and integration circuit rather than being sensory or motor in origin. We also found that this bias was present across all 4 task transition types, such that previous memories biased current decisions and vice versa. And we found that its magnitude in each context was correlated across individuals, both findings that are kind of consistent with a common source in a shared circuit for both functions. Along similar lines, we also isolated a general bias in our data toward particular spatial locations and away from others, and this is specifically toward oblique and away from cardinal angles. Just as with the history bias, we see that this general spatial bias was present in working memory and integration reports, but absent in immediate sensory motor responses, again suggesting an origin within a putative shared memory and integration circuit. And again, we see that the magnitude of this bias was correlated across participants on both tasks. So to summarize, with this work we uncovered convergent evidence from RNN modeling, neural decoding, and identification of these common sources of noise and bias and behavior that supports, I think, a shared circuit model for maintenance and integration of information over time, which, as I described, are these key computations for working memory and decision making. And for the particular cognitive tasks assessed here, the indication is that this shared circuit takes the form of a ring attractor. So to close, one exciting implication for us is that this kind of circuit model forms really these concrete links between levels of observation and analysis, from single neurons to neural circuits to manifolds and specific signatures in behavior. So really they hold promise not only for generating basic insights about brain function, but also insights about how specific loci of dysfunction can give rise to potentially multifaceted cognitive behavioral deficits. So this is the direction we're hoping to go in the coming years, and to get a look at our first attempt in the form of an application of our approach to cognitive aging, which relates nicely to the previous talk. I'd encourage you to reach out to myself or a stellar PhD student in the lab, Jade Duffy, who presented a poster on this work yesterday evening. So on that note, I'll close by thanking my collaborators on this work, and thank you for listening.
I think Steve was first, but I'm not sure. Go on then.
I was just wondering, so one of the predictions your network makes is this flat weights across samples for the decision task. So I'm just wondering, A, is that what you see in the human data? And more generally, if you had a decision task where you had a bound in play, how do you think the ring attractor would handle that kind of context?
Yeah, thanks for the question. I'm glad you asked. So we find actually that, sorry, there's a couple of things going on here. We find that in the humans, to answer your first question, there is not a flat kernel. There's some degree of primacy in the kernels. Interestingly, also, if we look at the reports in the working memory task of the humans, there's a slight attraction bias toward distractors, okay? So if you look at perfect imperviousness to distraction in the working memory task as one end of a continuum, and perfect integration on the integration task as the other end of that continuum, the humans lie somewhere in between on both tasks. So they don't reach either perfect end. But they do certainly adapt well across the two task contexts. Interestingly, in the RNNs, if we tweak the rule inputs, we can actually get them to essentially move between these two ends of the continuum as well. So these rule inputs seem to serve as these control signals that determine where the network falls along that continuum. In terms of how things would shape up if we impose a bound, I'm not sure. I mean, we'd need to look at that question, yeah. Thanks.
And quickly, Lawrence?
A related question on the rule inputs. So I guess you're showing really nicely that the generalization works at the level of the output of the network, and that it kind of nicely replicates things that we know about from handcrafted networks. But I wonder if you have asked the kind of flip side of that question as to how the network is integrating that rule-like signal, and shaping the connectivity, and whether that's in any way similar to what we know from handcrafted.
Yeah, that's like the outstanding question that we really want to address next, I think. We've got some candidate hypotheses there. Very briefly, I didn't have time to show this, but we've measured pupil signals on this task, and we show that these seem to be actually very intimately involved. They're very sensitive to task context, and particularly on the integration task, they're very sensitive to properties of individual stimuli that are presented and help, it appears, this network to integrate appropriately. So this could be one possible mechanism through which it adapts. Yeah.
Fab. Okay, let's thank Peter and all our speakers.
Thank you.
We're back at 5 for either Algonauts or the Climate Crisis Seminar.